Okay, welcome to Science Guy's coverage of Biology 1107. This will be the first workshop that many of you view, so I will go over just a couple basics as to how to use this course. We have workshops for all of the topics that your course will cover. There is a guide at the front desk that has the name of each workshop and all of the topics included, so if you're ever not 100% sure of what workshop you should be working on, you can check that guide. You can take a copy home and keep it if you want. So using these workshops, you should read through them and attempt the practice problems first and then watch these videos. That will give you a chance to engage with these ideas and then refine your understanding by watching the video. Unlike the chemistry courses, I'll mostly focus just on the practice problems in these workshops, just so I'm not reading a bunch of stuff to you that you've already read yourself. So for sake of time, generally I'll just focus on the questions, pointing out different concepts in the packet as needed. We will also, and if you're on the semester plan, this especially pertains to you, release a mock exam for each professor's particular course about a week, maybe 5 days before each exam. So you can look out for those as well. In the summer there'll just be one professor, but in the fall and spring there will be multiples. So make sure you're getting the mock exam for the correct professor because the material might be different. And lastly, we have study flashcards. There are several sets. Again, there's a guide at the front desk that will tell you which topics are in each set. If you're on the semester plan, you'll get all those and just use them as you need as the course progresses. If you're buying things individually, you want to make sure that you are buying the set that you need and understand that the more sets you buy, the more of a discount there is on the price. Okay, let's jump right in. So this workshop is, as it seems, an introduction to biology, so it will cover some basic scientific ideas and some basic ideas about living things. Most professors include at least a little bit of this stuff on their first exam, so even if it seems rudimentary, let's make sure we get those points and understand these fundamental concepts. So this workshop starts off with something, with the section about scientific thinking. It sort of describes what a scientific theory is, hits on a couple different types of reasoning here, inductive and deductive. These ideas do occasionally pop up in your curriculum and on an exam, so make sure you know the difference. The scientific method is here. Probably you've all seen this a million times. Probably you also know that real scientists will sort of poo-poo this because scientific research and discovery can happen in many ways, but this is a general idea of how sort of gold standard scientific research occurs. And then we run into our first practice problem here. So we're asked, just checking our understanding here about a scientific theory, because that's a concept that does show up on exams sometimes. Which of these is not a criterion for characterizing something as a scientific theory? A gives us it must have been tested multiple times and stood up to the results. Well, that's true. Excuse me, that's true. One of the justifications for a theory is that it must have been tested experimentally. It must have withstood multiple experimental tests, I should say. Withstood here meaning that the theory is supported by multiple experimental tests. Scientific theory is something that is going to have been tested multiple times. That idea that, oh, it's just a theory is sort of a misreading of what a scientific theory is. It's something that has withstood or stood up to many scientific tests, many different types of tests. So A is one of those criterion. B, it must fall within certain areas of research. That's not really true. So the concept of a scientific theory is not limited by the type of content, type of idea, type of research within which it falls. So a scientific theory can be about, I suppose, nearly an infinite amount of topics. So B is not a criterion. That is not a limitation to characterize something as a scientific theory. C, the third here, it must make testable predictions or claims. Is that true? Is that a criterion for saying something's a scientific theory? Yeah, it is. A scientific theory must be specific enough and sort of conceptualized in a way such that it makes predictions or claims that are testable. So a scientific theory, much like a good hypothesis, has to be testable. It must make claims or predictions that we can test, that other scientists, once the theory is conceived, so that other scientists can continue to test that. So A and C are examples of criteria that will characterize something as a scientific theory, sort of requirements, and B is not. So just make sure you read this little section on scientific theories and know the basics of what they are. Because again, it seems simple, but that can show up on an exam, and if you've ignored it, it might trick you. The next page here is about differentiating between living and non-living things. This also will show up on most professors' content at the beginning of the course. This is, after all, an important distinction. We sort of take for granted that some things are living, some aren't. But of course, in the nature of science, we meticulously develop definitions and requirements for categorizing things as this way and that holds true for living versus non-living things. I won't belabor the point here. You can read this on your own, but here you've got 1, 2, 3, 4, 5, 6, 7, seven different criteria for something to be considered living. So all of these things must be true. Importantly, all of them must be true for something to be considered alive. I've included here at the bottom that notably viruses are not classified as living. The reason is because they can't replicate on their own. You'll learn a little, most likely, depending on your professor, you'll spend a little bit of time on viruses, which is cool. Viruses are neat. Of course, they're scary too, but they're interesting. They infect cells and hijack cells to make them duplicate the virus DNA. They reproduce, but they have to sort of hijack a cell and use its machinery. On their own, viruses cannot reproduce, therefore they're not alive. But viruses do sort of challenge our understanding of what's alive and what's not, because they, in other ways, very much act like a living thing. So make sure you read through this page. Make sure you can identify not only identify something as alive or not, because you might just know that's alive. Make sure you can use these ideas to know why something is or isn't living. That way, if you get a multiple choice question about why is this thing alive or not, you can choose the correct criteria for what categorizes something as alive or not alive. We'll jump forward and we'll hit some practice questions about living versus non-living on the next page. Here we get to some basics about cells and organisms. So we're sort of creeping into the concept of hierarchy of organization, basically. So what I mean by that is we're getting into the concept of sort of emerging complexity, like the little pieces of living things that, starting all the way down with like atoms and going all the way up to organisms. And within there you'll have things like organelles and cells, and you'll get more complex all the way up to the level of organism. So organisms then are the smallest entity of life. Entity here meaning an independent living thing. So organisms are the smallest independent units of life. One living thing is an organism. An organism is a living thing. This mushroom is an organism. A human is an organism. A single-celled bacterial cell, we'll get into that in just a moment, is a living thing. All living things are made of cells. They're the smallest unit of life. So we're sort of contrasting entity and unit. Entity meaning a independent, fully functioning thing that is living, and unit here for cells meaning a small part of a whole. So cells are sort of self-contained units of life, meaning they are like little building blocks of life, but they don't function independently. A cell in a multicellular organism on its own cannot do all the things that categorize something as living. They are units, they are pieces of a organism. All right, so sort of complicating this idea is the fact that some cells, however, are organisms, because some organisms, some species on our planet, and perhaps others, are single-celled organisms, meaning they are only a single cell, but they are also the entirety of an organism. So some organisms consist of only a single cell. Most organisms consist of multiple cells. I list a few examples here of single-cell organisms, and as with all of these workshops, we're pulling from old exams and lecture slides and topic questions and whatnot. So these workshops, we try to, when there's a reason to, such as just like this is basically a memorization thing, it's very convenient to just sort of to be familiar with some different types of these single-cell organisms in case you come across a question that's asking you to perhaps identify all the organisms listed or whatever. So, you know, look at these, be familiar with them. You don't have to know everything about protozoa, or, you know, for example, but it's nice to have sort of a working memory of common single-cell organisms, and here are the ones that seem to come up in your curriculum. This is an image of bacterial cells here. Each one of these are an individual bacterium. So these are all individual cells and also all individual organisms. So that's an example of a single-cell organism. It's a single cell, it's also a single organism. Typically, a single cell is not an organism. In any multicellular organism, one cell that is a part of that multicellular organism is not itself an organism, but rather just a unit of a organism, a building block, if you will. Okay, a couple questions here, a few in a row. Is an E. coli bacterium alive? Explain. Well, E. coli bacterium. Okay, a bacterium means one organism of a bacteria, if you will. We know that most, or at least one cell of a bacteria. We know that most bacteria are single-cell organisms. It's in the section above, and that does include E. coli. So it's fairly common bacteria. It's one we want to memorize as indeed being a single-cell organism, bacteria. So if it's an organism, that means bacteria can carry on all the functions of living things. They meet all the standards for something being alive. So yeah, it's an organism, a single-cell organism, so it's alive, meaning it's indeed a living thing. It is alive. So I also noted here to make sure you can differentiate between common bacteria and viruses, remembering that viruses are not alive, because you very well may run into questions that ask you to determine if, you know, a list of things are alive, or if two things are alive, or if a single thing is alive. And it's very helpful to be able to differentiate between bacteria and viruses. That seems to pop up in curriculum where you might just be asked, you know, which of these two things are alive, and they're sort of assessing if you can say, oh, that's bacteria, that's alive, oh, that's a virus, viruses are not alive. So heads up for that. Make sure, again, as you're going through your course, take note of, in lecture and practice problems and readings, etc., take note of sort of common types of organisms, whether they be bacteria or common examples of single-cell organisms, common examples of viruses. Try to keep those things straight and ask yourself a lot, what type of organism or entity is this? Is it alive? Is it not? Just notice those things, keep them straight in your head, and that will help you. Okay, sort of on the same strand here, is an E. coli bacterium a cell or an organism? Explain. So here we're making sure we understand the difference between a cell and an organism, and also how they potentially cross over. So E. coli, we established, is a living thing. So kind of right off the bat, hopefully we'll say, well, it's an organism because organisms, organisms are a single entity of a living thing. E. coli itself is a living thing, therefore it's an organism. But we can reason a little more in a more granular fashion. A cell is the smallest unit, so it's not on its own a living thing, but just a functioning part of a living thing. Organisms are full living entities. We know E. coli is alive. We know it's a living thing. We established that. So a single bacterium, remember that means a single bacterial cell, of E. coli must be an organism. This is true. So that's one way to answer this question is, I think the best answer here is that it's an organism, because that's sort of harder to be categorized. But remember, you could also sort of say both here, right? Because a E. coli bacterium means a single E. coli cell, bacterial cell. And we know that E. coli is a single cell organism, so it is both a cell and an organism. It is a single cell that functions as an organism because it satisfies all the requirements to be categorized as a living thing. So another way to answer this question is both. If you had to pick one, you should go with organism because that's more specific. But you can definitely answer this question by saying it is both a cell and an organism, and explaining that single-cell organisms are fully independent living things that also happen to consist of only one cell. Okay, let's move on to page 5. Continuing with some practice questions here. So we're asked now to compare and contrast a bacteriophage, which is a virus that affects bacteria, and an E. coli bacterial cell. I'll throw out there that this is a good word to sort of memorize what it means. Phage is a word that's associated with viruses. It's sort of one virus, one unit of a virus, a single virus cell if you want. It's not really a cell, we call it a phage instead. If you see this word, we're talking about viruses. That's good to know. Okay, so comparing and contrasting an E. coli bacterial cell and a bacteriophage. So these, there are some similarities we can draw, but mostly they're going to be very different because they're two complete different categories of organisms. Bacteria, which are generally single-cell organisms, and viruses, which are not alive and sort of feast on living things or cells. So for instance, some of those differences, a virus is not alive, as I mentioned, whereas E. coli and other bacteria are living organisms. Not all bacteria are single cell, but all bacteria are alive. Because of this, bacteria can reproduce on their own and in multiple settings, whereas viruses can only reproduce, as I mentioned earlier, using the machinery, so to speak, of a cell. It's also a big difference in size that's notable and might potentially be in your curriculum, because in this introductory stuff, as we'll see on the next page, there are questions sometimes about relative size of different types of molecules and things. There's a big difference here. Bacteria are generally much larger than viruses. Viruses are real small. Bacteria cells usually much, much larger. If you wanted to draw some similarities, they're both microscopic. They can both cause disease to multiple organisms. But even that being said, they infect their hosts differently. A bacterial infection is different from a viral infection. You probably know that by the concept of just like medicine, right? Antiviral drugs versus antibacterial drugs, and one will not help to heal you from the other. You can't use antibiotics to help rid yourself of a viral infection. So they infect differently. Okay, let's continue. Scientists generally accept the scientific theory that mitochondria are sort of a result of an ancient bacteria cell being engulfed by a eukaryotic cell. So the basic idea, without getting in the weeds, is that there was a symbiotic relationship between some types of eukaryotic cells and bacteria cells. The bacteria cells were sort of swallowed by the eukaryotic cell, and that helped the eukaryotic cell survive and reproduce. Over time, intermingling of DNA would happen such that when those eukaryotic cells reproduced, they reproduced the engulfed bacteria cell, and over time that bacteria cell evolved to function as a mitochondrion. So which would be, given that theory, which would you expect to be larger, a mitochondrion, a single mitochondria, or a bacterial cell? Explain your reasoning. Well, kind of just for visual learners, adding a little visual here. If a mitochondria, this is sort of a before, ancient before, modern after, if you will. So if mitochondria are the result of a prokaryotic bacteria cell being engulfed by a larger eukaryotic cell, we should expect the size of a mitochondrion in a bacteria cell to be similar if this mitochondrion evolved from this type of prokaryotic cell simply being engulfed and eventually becoming part of this eukaryotic cell. We'd expect them to be similar because this evolved from this, so we should see a lot of similarities, vestiges from its past. So we'd expect the size to be similar for that reason. So that's some basic reasoning there. I'll go a little farther because this concept pops up in some professors' material. It's also interesting. It's also true that mitochondria, and this is true of chloroplasts as well. It seems that chloroplasts in plant cells that do photosynthesis are vestiges of engulfed bacterial cells as well. They would have been photosynthetic bacteria that were absorbed and evolved, again through symbiosis and DNA exchange, into chloroplasts that now nearly all photosynthetic plants have as an organelle that helps them undergo photosynthesis and, of course, create energy from the sun. So I always think it's worth remembering that some photosynthetic bacteria are the only reason that we exist. So thank a tree today. So it's true that mitochondria and chloroplasts seem to have come about this way. They also have striking similarities to bacterial cells. They both have their own independent, separate independent DNA. They produce proteins and enzymes required for their own function as if they were an independent entity. And other details. You can read these on your own. Strikingly, both those organelles reproduce in a similar way to bacteria. They replicate their own DNA and direct their own division. So it very much seems like they, in the past, were their own entity. So just tuck that into your mind and understand that about mitochondria and chloroplasts. Also be able to reason in this way. And this next question sort of piggybacks on that and asks us, what type of reasoning did we just use? So here we're assessing, do we understand the difference between inductive and deductive reasoning? This was deductive reasoning because we're provided the premise that mitochondria evolved from bacteria, and then we drew one conclusion in this case about their relative size. So remember, so you've got, if we sort of set two things apart here, specific details and a general premise or idea, you can think of this as like a theory and the predictions that would stem from that theory, or different observations and a unifying idea that unifies them. So inductive reasoning is when we have a bunch of specifics and from that we draw a more general conclusion or premise or idea. And deductive reasoning is when we have a general premise or idea, theory, what have you, and from that we predict or assume different details. We go from that general premise and apply it in specific ways, if you will. So lots of details going towards a more general idea is inductive. A general idea going towards more specific ideas is deductive. So make sure you understand that in case you see a question about it. It's also interesting to think about how we reason, what we're doing with our brains in the day-to-day. Okay, finishing up here on page 6. I mentioned this a little bit earlier, hierarchy of biological organization. So we're basically talking about going all the way down to the most minute and granular level of life, talking about the atoms that make up molecules that make up the tiny structures within cells, all the way up to fully functioning organisms. And then if you're talking about ecology, you can go all the way up to groups of organisms, populations, and eventually the biome, all of the life on Earth. Alrighty, so What I've done here is given you in this image the basics within, like the human body, or the basics in an animal. This is real animal and human centric here, but it's helpful. Also, what I've listed here is drawing from a bunch of different exams and lecture slides. This is a smattering of different types of molecules and structures within organisms that you may need to know about, such that you can discuss their relative size. So I would encourage you to essentially memorize this, think through this, try to envision it in your head, and get, because we may think, and many of us probably do, but some of us may simply think that we understand the relative size of these fundamental structures, but you can get into gray areas pretty quickly. So I think spending a little time and getting to where you can sort of, in your mind, take a tour through these types of structures and see the relative size can be very helpful. I explained what the slashes and the arrows mean here. I'm going to point you towards a particularly useful multimedia tool if this gives you any trouble and you need to know it. If you sort of just Google or whatever cell size and scale, one of the top options is going to be this page from Utah University, I don't know, their genetics department. And it is a cool visual where you basically slide a bar from left to right, and it goes from macro to micro. It sort of dives into smaller and smaller structures, and it shows you relative sizes along the way. So that could be helpful. So use that in this material, and again, note the patterns and the repeating structures that come up in lecture, and make sure that you can compare and contrast, compare their relative sizes, because there's a high probability you'll have to do that on a question or two on an exam. So last couple practice problems here. Engage with this. Order the following structures from smallest to largest. And here is my list. Based on just what I've given you here, we know that single amino acids and nucleotides, which are the functional units of DNA and RNA, nucleotides are. You'll learn more about that when you get into macromolecules, but they're just small little pieces, little single units of DNA and RNA. They're quite small. That is the smallest thing that we have listed here in our list. Here is the DNA nucleotide. It's a nucleotide. It would be about the same size as an RNA nucleotide, for the record. And then a little bigger than that are secretory proteins. That is on our list up here. Large proteins being a bit bigger than nucleotides. Secretory proteins are generally going to be large proteins. You don't know that yet, but it is true. And we see that viruses are a bit bigger than large proteins in our list. So the West Nile virus, of course, is a virus, so it's next. Lysosomes are large organelles. Mitochondria are also large organelles. So it being one of those big organelles, it is larger than a virus. You'll note here that large organelles, viruses, and small organelles are kind of in a row here, so you might want to take note of that. And lastly, an amoeba cell, because an amoeba cell, of course, is an entire cell. While it didn't matter because there was not a eukaryotic cell around, or rather, not a multicellular organism cell around. Amoebas are on the list of single-cell organisms. Note also that they are often used to represent a eukaryotic single-cell organism. We typically associate single-cell organisms with prokaryotic cells, the simpler and more ancient type of cell. You'll get into that in the Cell Basics workshop. But amoebas are single-cell organisms and eukaryotic, so they are a common example of that. So that's worth also tucking in your brain bank there. So yeah, the amoeba cell, regardless of what type of cell, is definitely going to be the largest, because we need to understand, and here's a general takeaway from this, nucleotides are small molecules that are part of DNA molecules, which are larger. Proteins are a macromolecule that can be big or small, but that, it can take a bunch of proteins to make up small structures within a cell. Viruses are also small things that can affect a cell. Then lysosomes, you're getting into like a bigger structure within a cell, a larger organelle. But all of these things, and we need to be able to visualize this in our head, are small parts within a cell. So the cell is going to be, any type of cell is going to be larger than these things, which are components of a cell. In the same way that, bridging to the next question here, organs are small things, relatively speaking, that make up an entire organism. Organelles in the same way are small things that make up cells that allow cells to function. Organelles inside cells, organs inside organisms. So that being said, that takes us to our final question here. In several sentences, explain the progression from less complex to more complex of organelles, cells, tissues, and organs. So hopefully we can, without too much trouble, and if we have trouble with it, no big deal. That's why we're watching this video, right? We're able to realize that organelles are within cells. They are functional parts that allow cells to work. That many cells make up tissue, and that collections of tissue are what make up organs, knowing also that organs are sort of the organelles of an organism. They are the functional specialized structures that allow an organism to do all the metabolic things it needs to do to live and reproduce. Okay, so this is how I would explain this. Of course, this could be explained in many ways, but I think these are the basic ideas. We need to know, I've already mentioned, drawn the parallel between organelles being like the organs for the cell. Organelle, organ. So organelles are specialized, and they carry out specific functions in a cell, allowing the cell to carry out all its functions. They therefore sort of make up the basic units. Cells that have to carry out all their own functions and sort of keep themselves functioning, they together are the functional basic units of an organism. Yep. So every organism is made up of tons of cells. But it's not just a bunch of cells organized all willy-nilly. Cells, different types of cells, are organized, sort of compartmentalized, such that large groups of cells with common characteristics, the same types of cells, they come together, they aggregate and form different types of tissue. So you have all sorts of different types of cells, and you get a bunch of one type of cell together, and they communicate with one another, and all of their independent functions lead to the creation of what we call tissue, which is a collection of specific types of cells. And then that tissue, in the more macro sense, takes on the properties, the abilities and limitations of those cells. They work together to do like a larger, more complex thing. And then the complexity continues to increase where tissues, and sometimes multiple types of tissues, oftentimes, they aggregate and they form these functional things called organs. So like, your heart has specific types of tissues so that it can carry out its function, so that it can beat, moving blood through your body, so that it can communicate with your nervous system, etc., etc., etc. It needs different types of tissues to do what it needs to do, and those different types of tissues have specific properties that stem from the fact that it is made up of a bunch of specific types of cell. Then organs are for the organism what organisms are for the cell, specific functions that allow the organism to carry out all its complex functions. So that is the degree to which you need to understand sort of how we emerge as complex organisms all the way down from the small organelles within cells. Of course, we could go deeper. We could go down to macromolecules like carbohydrates and lipids, and even there we can go down to the chemical functional groups, and even down to atoms, and then to subatomic particles, and even further from there. There is I mentioned functional groups there. You'll move on to a workshop that outlines all the chemistry that you'll need to know for this course, and then to some cell basics, and Also, pretty soon, and generally in this unit, you'll move on to the workshop with macromolecules, carbs, lipids, nucleotides, and proteins, and learn all about those. So don't fret if some of this stuff is sounding like I've heard that word, but I don't know anything about it. This is just the introduction. You'll get into a lot of these specifics. Okay, so we went through what, like I said, probably seems like review, or at least things we're familiar with, but this workshop draws on material that has shown up in the past. So hopefully this gets you oriented to how you need to be thinking about these fundamental ideas. Thanks for studying with us, and have a great day.